Hello, everybody, and welcome to our next class. You have now, in the previous class, just run your first ChIPseq analysis, which is fantastic. And so now what we want to do is give you some background of what all was run, what programs were run, and the pipeline that Nextflow put together for us. And a little more background about what the biology we're going to start to explore is, just to sort of give a context now that we've run the data, what are we looking at, and get back on the same page. Towards our goal of doing a much larger ChIPseq data set. So you guys have done all of this now, and we are ready to take another level in and see what we're looking at here. And so as we're going to explore the human genome, just to give a little bit of background, that genes can be defined by a promoter region where the DNA binding proteins that we're analyzing the ChIP data for, or that you just did analyze the ChIP data for, all bind on a region called a promoter region. And that's marked by this histone mark that packages DNA that we'll talk more about, and we downloaded this data as well. But you can tell where a start of a gene is by this lysine 4 trimethylation of a histone versus lysine 36 trimethylation on the same histone indicates the polymerase has gone through. And this is just indicating a gene down here that has exons that are spliced together to make a full transcript. And these are the sort of loci we're going to be looking at throughout the class and see what DNA binding proteins come here that might initiate this process of activity of this gene. So what we're going to see is tracks that look more like this. Nextflow's already produced them for you, and we're going to go look at those in visual browsers in the next class. But for now, there's some data shown here where you can see a promoter and then the transcribed region, promoter, transcribed region. So you can almost see where the genes are just by these two DNA binding proteins, which actually are the packaging histones of DNA. And we're going to look at dozens of DNA binding proteins that might be binding in this region to regulate this gene. Okay, so this could either be, this signature is the same for an mRNA that's going to be translated into a protein, or an RNA that would look just the same but acts as an RNA instead, which is called a long non-coding RNA, or lincRNA for short. So this ChIP data that we've just analyzed will allow us to see where promoter regions are, and then later we will get expression data to start to look at what is being activated or repressed at these sites. So just a little bit about regulation here, that you can have activators and repressors. We found some repressors in last year's class. And so there's the combination of DNA binding proteins. This is just 3 shown here, and we're going to look at over 100 of them. So we'll get a much larger combinatorial picture. But this is all in the promoter region where these guys will bind and either repress or activate depending on which combination actually ends up there. And that's sort of outlined here from a cartoon that is referenced in the link below. So this is called ChIP, or chromatin immunoprecipitation. And you want to look at the genome-wide pattern of these DNA binding proteins. You can think of it as playing the keyboard, that different fingers on the keyboard are going to make a different note, and different DNA binding proteins on the promoter are going to make a different output of the gene. So this is just sort of showing that usually you have transcription factors, which are a type of DNA binding protein that land on this promoter. And then this is the polymerase, which is the working part of this activation, and it transcribes the gene itself. So it alone can't do it. It sort of needs an activator to push it along. And these are the sort of proteins we're going to be looking at, or a repressor that prevents it from moving along as well. And so there's 3 types of these polymerases that make the mRNA, or long non-coding RNA transcripts. RNA polymerase 1 tends to make ribosomal RNA. Polymerase 2 does long non-coding RNAs and mRNA. And polymerase 3 does more RNAs that are involved in translation. But pole 1 and pole 3, it should be noted, we're only going to be really looking at pole 2 regions, which are the mRNAs and long non-coding RNAs. But the majority of the RNA that's synthesized in the cell is made by pole 1 and pole 3. And this won the Nobel Prize twice by a father and son duo. But just to note that we're only looking at a small subset of the total activity in the human genome. Promoters can be complicated. They can show up anywhere. So you could see here's a promoter of a protein coding gene. Here are the exons that are going to be spliced in blue, and the protein coding gene goes this way. If there could be another promoter on the other strand of DNA that makes an RNA that goes in the opposite direction. That's called antisense. A gene can be contained completely within an intron of a gene, and that's called intronic. And bidirectional is when two genes share the same promoter, and one gene goes in one direction and one gene goes in the other. And then intergenic is just referring to, or intervening is just referring to non-coding RNAs that just sit in between protein coding genes. And this doesn't really matter until later in the course, but we are going to be mapping to all of these types of promoters and looking at how they might be regulated differently. And so to sort of put together the three aspects we just mentioned is that we can get multiple layers of evidence when we have a promoter and a gene body here by histone packaging, how the DNA is packaged into a promoter versus the body of the gene. Also looking at its coding potential, whether it's likely to be a protein or not, and an RNA map underneath it. And we'll sort of make all of these layers plus another layer of all the DNA binding proteins localized at these promoters. So if you take one of the most powerful transcription factors is PPR gamma, where you can put this transcription factor into a cell and turn it into an adipocyte or a fat cell. And so it's quite powerful that if we can know where these DNA binding proteins are, we can understand what genes they're turning on and regulating and how that defines different cell states. In this case, we'll be looking at the HepG2 cell state. And this comes back to this combinatorial aspect of DNA binding at promoters that we discussed a minute ago. And so just to refresh on CHIP, what we're looking at is DNA binding proteins are just defined in this case as something that is close enough to DNA that when you pull on it, you retrieve DNA from it. So in this case, what you do is you cross-link cells and freeze frame them with 1% formaldehyde. You lyse the cells, so they're all in place. Everybody's just where they were before. And you take your protein of interest and you IP it, and in the input control, we've downloaded and analyzed in our first IP, and we'll look at that in the next video. But this input DNA is really important so we can compare how much more DNA we get when we target our protein of interest versus random DNA that's pulled down. And then we chew away the protein and RNA and we sequence the DNA. And what we're going to be looking at is raw data that looks like this, where the y-axis is simply the number of reads that show up. And in the IP, you'd hope there's more reads in a region, such as a promoter, whereas input DNA will have some structure to it, and we need to be cognizant of that, but much less than the IP. And so what we're going to go through is each of the steps that the pipeline pumped out, but this is just sort of getting closer to what that is that the FASTQ files are simply a bunch of reads. We align them to the human genome, and you can see them starting to clump up here. And MAX, which we'll talk about, is one of the programs run in the Nextflow ChIPseq pipeline. We'll start to say, oh, I see more signal here than I do here, and it'll give you these nice handy squares where it says, I see a signal in the genome here and here and here. It'll also give you these tracks too, which are called bigWig files, which we'll get into more a little bit today and more in the next video when we do visualization of the data. That's sort of the process that the ChIPseq pipeline did, is it took your raw reads, aligned them to the genome, constructed signal, and made peaks, and tells you where the signal is. Just a little bit of background on sequencing, since we're dealing with sequencing data, is what you basically are doing when you IP your protein is you adapt sequences onto the ends of it. So you have your protein bound to DNA, you've IP'd that, you shear it, and you cut the ends off, and then you ligate on adapters. And this is important because this allows it to attach to the glass surface on both sides in this adapter, and then also a sequencing primer that can be read in both directions. So you can sequence a single end read, you would sequence just in one direction, and in a paired end read, you'd read in both directions. And we'll get more into that, but the data we're looking at is just single-ended, so we're just going to read this sequence straight through until we hit the next adapter on the other side. And you'll see why these adapters are needed is that they're put on there so that they land on the glass, and then after they're replicated, the other adapter is going to now stick to the glass and then be released after the next round of replication. So what you end up is a cluster of the same sequence. So your initial one piece of DNA that was represented in your chip is now being amplified into many versions of that single thing in one spot. You can think of that as a colony on a bacterial plate. Is very similar to this one piece of sequence being cloned and amplified in a single position on a glass slide. Since it's on the glass side and you have these immobilized surfaces, this adapter here is going to then hybridize here to make these arches, which are replicated into these clusters. And this is just more on that, but I pretty much described it in the first one. And the key is that these adapters are going to have to be taken out before we align to the genome. And we'll see how that's handled by Nextflow as well. And what's cool is that on this glass slide, now that these sequences are mobilized and amplified, you'll start to see as one letter is put in, let's say a T, it's going to match with an A, and you're going to see a blue thing on a blue color. And now you know that the next letter is a T. You wash that out, you add another letter, and then you see what letters show up here in sequence. And then by doing that, you're essentially saying, oh, I first saw a T, and then I saw a G because it was green, and then I saw a C because it was orange, and then I saw a T again because it's blue, and A again because it's purple, and a C because it's orange. And so it just goes on in concession. And nowadays, 150 base pair reads are perfectly fine on both ends. And this allows you to have a 1,000 base pair sequence. And if you read 150 base pairs from each side, you can infer the linear sequence that was in between there, or gaps that were in there if there was a deletion in the genome, etc. Okay, so we can do this. Here's a typical ChIPseq pipeline. We're going to get data that looks like this that we'll look at in the next video. But right now we want to show you what Nextflow did for you. It did all of this stuff, and it gave you so many great files that we're going to be able to go look in the next video and already look at the data, where the peaks are, and get ready to do more statistical analysis coming up. So the first thing we want to do is FASTQ, make sure that our reads are of not all the same, like not all A's, not all G's, not all T's, and how well it called that color in the circle, sort of a quality control if you will. And that's the first step, is FASTQC. If you were following along with the tail of the Nextflow output, you saw this was one of the first things run. It's just basically saying what the quality, how well the quality of your bases were at different read lengths. And you'll notice that if you have homopolymers of all A or all T, G, C, you'll see really bad quality score metrics because the camera can't read that many dots at once. But you can dig into this more. This is just the first step, which says, hey, how do your reads look? And can I align them? So then we want to trim those adapters we had put on that we had just mentioned to help get it on the glass and sequence it. This is done in Perl. So I'm highlighting this because Nextflow can use all of these languages, all of these different packages that are made by different people and have different versions. And it's a package manager that knows to go get all of these things and get the latest version of them and put them in sequential order, and make sure that you know, or if somebody wants to reproduce your data, it'll give you all of the needed information of what was run in this pipeline. So we take those out, and this is the most important part, is aligning it to the human genome. This has been a complex aspect for a long time. It used to be that the reads were less than the genome, and therefore it was best to JPEG, if you will, or collapse your read sequence complexity. And now we get so many reads that somebody decided to just let's index the human genome. And so the Burrows-Wheeler alignment is worth lots of studying. I think it could deserve its whole class in of itself. I'm just going to give you a high-level conceptual aspect of it. And there's 3 different versions that Nextflow will run depending on the read length and whether it's paired end. And other applications such as RNA-seq, you might be using different versions of this, but Nextflow will know what to put in each pipeline. And so the input are these FASTQ files, which are the raw sequence reads that now have the adapters trimmed off so they can be aligned to the genome. And that might seem daunting. How do you take a sequence read and find where its matches in the human genome? And this is just a simple example of a Burrows-Wheeler's transform of banana, where this caret means first letter in regular expression. And so what you can do is say, here's all the possible ways you could do banana. You could start with just the whole word banana. You could start with a space and then banana, and then you could start with B-A-N-A, and then your last letter would be an A. It would know that the last letter was an A. You could start with this, sort them. I don't want to go through all this. It's way too complex and way too smart for me. But what you get in the end is a quick way to index this. So if you see BNN, then you know that there's an A in between each one, and that the last letter is an A, and then you get banana. And so it can search only 3 letters that sort of compress this, if you will, like a JPEG picture is taking all the redundant pixels and shrinking them. Essentially, that's what Burrows-Wheeler is doing to the genome, is just shrinking it and making it also accessible and indexable. And so just to give sort of a conceptual example of that, if you had a short read here, and what's interesting is you have to do this in one direction. It doesn't work in both directions. So we're going to go backwards and say our first letter is a T. And so we say, oh, all the T's are down here from that index we just looked at. And then the next letter is an A. And if you have TA, you're somewhere in this region. And then if you have TAA, you're here. And then it can hone in quite quickly after the next few reads that you're right here. Then it finds where you are. And it doesn't need to use the whole read to know exactly where you are. And this is what the output of that is. So the Burrows-Wheeler is going to say, here's where your sequence is, and here's where it matches in the genome. This is very hard to read, but it is actually readable. These are called cigars that are telling you essentially where the read is. You don't have to know all of this. You can Google it all you want. But basically it just says, here was the sequence read with the adapter removed, and here's where its position is in the human genome, with a bunch of complicated ways of doing that. So this is a SAM file. You can actually read it as a human. BAM was then quickly adapted in binary to compress this about 4x size. These are large files. You can imagine there's millions of reads, and they're typically about a gigabyte in size. So having 4x compression is really good with BAM, but you can't read it. In SAM, you can. And then we'll look at, we're not going to go into it, but SAM tools and BAM tools are being used ad nauseum throughout this pipeline. And here's just an example of Picard and pre-seq, which is going to get rid of all duplicates, split reads, and mismatches for ChIPseq at least. Sometimes you might want split reads, which means that one end has a gap in the linear version of the genome, but then it like splits into another site, and that can be a sign of splicing of RNA. But for Chip-seq, you wouldn't expect any of that. So one way you could have really bad data that got to this point is that if you amplified, let's say, one piece of DNA and it just jackpotted and you just got the same sequence a million times, this Picard and pre-seq are going to get rid of that. And that happens a lot with ChIP-seq other types of seq where you get finite amounts of stuff and you do this massive amplification. You can get if you get a lot of duplicates of something, it's probably a PCR artifact. There's ways of putting barcodes on so that you know that it's not a PCR artifact, so that every adapter has a unique barcode. So if you see the same barcode over and over again, you know it's a duplicate artifact. But for this level, we've now gone from aligning the reads, and now we're going to get rid of all the bad sort of stuff that's happening. And this, again, is just to say SAM tools and bed tools are being used back and forth in many different ways to take those those alignment files, which are kind of the most raw aspect of this step moving forward to calling peaks. Deep tools is going to tell you how well your data sort of looks for calling peaks or what its statistical significance is. And so what Deep Tools is doing is very cool. It's called a plot fingerprint. And remember that mark we talked about just a little bit ago, this K4 lysine 4 trimethylation histone, which is at promoters. It's a very sharp peak. And so peaks come in different sizes. They can be sharp, they can be broad, and you do want to keep this into consideration. In Deep Tools, you have this in your folder already. But what you'll see here is that there's a large gap between input and when you hit sort of saturation, which means they're very sharp peaks. Similarly here, what you see is that this arc is going to take a lot longer. You can read more about this. I just conceptually see that this is an indication of a narrow peak, and this is an indication of a broad peak. And this is an indication where your chip signal is not much more than input, meaning that basically it looks like total genomic DNA, which could mean your protein binds everywhere in the genome, but the chances of you calling peaks are not going to be very good because no reads will stack up because it looks like the entire genome. Okay, so that's Deep Tools, which is sort of giving you some insight already into what kind of signal you're looking at. Picard, we already just talked about, gets rid of that. And then Phantom Peak Tools is going to say, take another level and not only look at that plot profile, but look at what how the reads are aligned on strands. So you would expect that the DNA binding protein would be binding sort of to both strands, and you would see equal representation on either side of its binding site. And that's sort of schematized here. And so what you would expect is something like this. It's not usually as perfect in a typical ChIPseq, but this would not, this would be considered low complexity. So this would not be a duplicate, so to speak, that Picard would have picked up. But this is saying, hey, look, this doesn't make sense. We expect that there'd be some average shuffling between the sense and antisense strands. And so Deep Tools is taking that into further consideration. And so just as a final thing that Max is doing is going to take a window, a narrow window and a broad window, and I'll sort of represent that. But I'm going to represent this with 3 windows, which actually could be many windows, which is called a scan statistic. I just want to highlight this of why this would be important. That is, we've run a small window. We would probably see this isn't significant. Imagine each of those arrows is a read in the ChIPseq. No, it's still not significant. Okay, so now it starts to get significant. Yep. Okay, and then no, our read signal goes down over input. Okay, that's what a small window would do. But now a medium window would then say, oh, the signal's more in the center, and a large window is only going to get that only one little spot where it's significant. And this is important because if you have a sharp window, you want to use small windows so that you get that quick up and down of the peak. And if it's a long one, you want a large one. Max is going to run 2 of them and do all the right statistics that are fantastic. But you could develop a scan statistic to add to this where you test many different window sizes and say which window gave me the most significant coverage. And then that would fit the shape of your peak a little better. Bit esoteric, a little too much for now. But for now, just know that you're going to get broad peak and narrow peak files back out from Nextflow. And that's kind of this concept here where it ran a narrow window to see where the sharp peaks were and a broad window to find where the broad peaks were. So what does this all look like? We're going to see tons of these tracks. This is the next video. We're going to start looking at these already from the run you just did. But you can compare two cell states really easily when you see where this DNA binding protein is. So you can see that, oh, this gene is off, these two are on because it's bound there. But in this cell state, now you see that it's gene 4 that's only on where this guy is bound. And you'll see so many of these tracks, it's going to start to make more sense. But really what we're looking at is how DNA binding proteins are organized around the genome. In this picture, we're looking at 4. Imagine looking at over 100, and that's our goal for this class. Ultimately, right now we've only looked at 1, but it'll still be informative, and we'll see these patterns already starting to emerge around the genome. And eventually, something like this will make sense to you of how DNA binding tracks change. Here you can kind of see a narrow peak versus a broad peak, and RNA-seq underneath these peaks. And I don't expect you to understand any of this at this point, except for these are the kind of tracks we're going to start to look at in the next video to see how well does our data reproduce. How do our replicates look the same? What happens if we run it on 2 different versions of the Nextflow Chip-seq pipeline? Do I get different peaks? Some of these concerns, just a very quick overview of data visualization in IGV to start looking at your data that you've just generated. So let's go look at the directories of what NFCore has produced for us, and a lot of these files that we can then throw right into a visualization browser and start looking at your data. So congratulations, you've run ChIPseq, and now it's time to look at it. In the meantime, code light.